Wonderful. We're on. All right. Welcome, everybody, to the 6th edition of the Multiomic Reading Group. I'm your host for today, Cesar Miguel Valdez Cordova. And today we have a very special guest. And I would also like to mention that it is our very first speaker who attends in person. We're going to thank him in a minute after the introduction is made. But before that, we'd also like to thank us being the organizers of the Multiomics Reading Group, David Amelin, Quentin Baron from the BioAI Reading Group, and Quentin Fournier for contacting the speaker and making this all happen. So I'll introduce our speaker today, Thomas Pierrot. He earned his PhD in deep reinforcement learning from the Sorbonne University in Paris. Throughout his doctoral studies, he focused on enhancing exploration and sample efficiency in deep reinforcement learning and collaborated with Google DeepMind and Imperial College London. Since completing his PhD, Dr. Pierrot's research interests have transitioned towards developing representation learning and generative methods for biology. He currently leads the genomics AI team at InstaDeep, the AI arm of the BioNTech group. BioNTech is a German biotechnology company that develops and produces immunotherapies and vaccines for various diseases, notably known for their mRNA-based COVID-19 vaccine developed in partnership with Pfizer. InstaDeep's genomics AI team is composed of 15 researchers spread across Paris and Boston, with a mission to develop the next generation of biofoundation models to accelerate innovation in the field. Let's take a minute to welcome our speaker, Dr. Thomas Pierrot. So for a little talk introduction, there's been some papers circling online. There's been a lot of hype around the talk. I'll give a little introduction to it. The human genome sequence provides the underlying code for human biology. Since the sequencing of the human genome 20 years ago, a main challenge in genomics has been the prediction of molecular phenotypes from DNA sequences alone. Models that can read the genome of each individual and predict the different regulatory layers and cellular processes hold the promise to better understand, prevent, and treat disease. Here, we introduce, or rather, Dr. Thomas will introduce the Nucleotide Transformer, an initiative to build robust and general DNA foundation models that learn the languages of genomic sequences and molecular phenotypes. NT models ranging from 100 million to 2.5 billion parameters learn transferable, context-specific representations of nucleotide sequences and can be fine-tuned at low cost to solve a variety of genomics applications. In this talk, he will share insights about how to construct robust foundation models to encode genomic sequences and how to evaluate them. We will also present recent advancements of the group, including a study of the performance of such models on protein tasks, as well as ongoing processes and progress towards more general genomics AI agents that can integrate different modalities and show improved transfer capabilities. The training and application of such foundational models in genomics provides a widely applicable stepping stone to bridge the gap of accurate predictions from DNA sequence and makes a step towards building a virtual cell. Without any further ado, Dr. Thomas Pierrot, please take it away.
Right. Thank you so much. So can I move to the presentation? Yeah. Was it on a different tab? That one? All right. Is it good?
Yes.
Great. Thank you so much. Thank you so much for the introduction.
Actually, I don't think you see the new slide on now.
Oh.
Well, sharing screen, but
All right.
I wonder what's going on. I'll just put the link on the other window. This should do
Now
it.
we can see the screen, but I think when you shared it.
On like the slideshow.
Okay, no, that's good.
It's good?
good?
Okay,
Yeah.
wonderful.
Right. Right. So yeah, thank you so much for the invitation. I had the chance to come to Montreal a few times before, never to Mila. So I've heard a lot about it. So it's pretty cool. Always nice to have the opportunity to spend the weekend in Montreal. I live in Boston. Always nice to be able to speak French for a few days. So today I show kind of part of our research roadmap in our group at InstaDeep and BioNTech, and I'm mainly focused on everything around building foundation models in the specific context of genomics for this talk. And I just wanted to start with a definition because in the field, many people talked a lot about what it means to build language models, foundation models, large language models. And so For us, we can't use this very generic definition saying that any foundation model is going to be one large AI model, a deep neural network, usually transformer, for instance, that's been trained on lots of data and that can be used to perform a wide range of tasks. And then you can train these things in lots of different ways, and I show several during this talk. You can use unsupervised learning, self-supervised learning, next token prediction, mask language modeling, there are lots of techniques. You can also use supervised learning. It's a bit less the case in some fields, like in proteomics, but for instance, in the case of genomics, there's been some models like Isoformer by DeepMind, or recently the new version, Borzoi, that's been trained purely through supervised learning, but on data sets that are so big that they actually also run very good representations. And in our group, we asked the question of how can we take all the data that's available in genomics. And so in genomics, there are actually lots of data when if you compare to English. So there are data sets like NCBI with genomes from lots of different species, usually reference genomes. There are data sets like the 1,000 genomes with genomes of different individuals, so also of different human genomes, but with mutations. And then there are a whole bunch of data sets like encode, GTech, where you're going to go look into cells and try to understand how this genome is going to be transcribed, look at the presence of mRNA, etc. To give an idea, already just NCBI, you have a few trillion tokens. If you start adding data sets like encode, you multiply almost by 1,000 the amount of data. Right now in the community, I think we have only scratched the surface. And so a bit the idea of this research is to open all these things together to kind of build models that start understanding the fundamental biology of the cell, and maybe start, and I give a few words about that at the end of the talk, start building even agents that we can use to interact with and ask questions. I focus on 4 different directions for this talk. So I start from the Nucleotide Transformer, so basically how to learn from genomes. So I just have lots of genomes. Maybe I have no annotations, I have no labels, but I know they exist, they've been sequenced in nature, and I want to learn from that. And then start building from there and see, can I add other modalities? Can I try to understand what's going on inside the cell? Can I add the presence of RNA, protein? Can I start adding annotations of the genome? And finally, can I even add the language to the mix and see if I can make these things conversational and start asking a question? So this first work, the Nucleotide Transformer, that's just been published in Nature Methods a few months ago now. We started a long time ago. It's been a long review process, almost more than 2 years ago, with this idea that's been used now lots of fields, also lots in science, especially in proteomics, on how to use mask language modeling to run representation. And so the way we started this study, that was just like, okay, let's scrape all of the genomes that we can find, typically in NCBI, so let's take genomes from hundreds of different species, and start cutting these genomes into chunks, because usually genomes can be very long. So human genome is 3.1 billion base pairs. So you're not going to fit that directly in a deep neural network. So we started to cut this genome in pieces, started to tokenize, press with lots of techniques. We ended up tokenizing with 6 MERS. I'm happy to take questions. There are lots of interesting work on what's the best way. But for us, it was kind of a good compromise. And then started to use mask language modeling. So you take some of these tokens, you randomly drop them, and you train the neural net to reconstruct what's been masked. And we played this game for quite some time. Looked at lots of different assumptions. We looked a lot at the effect of the pre-training data set, starting from just taking one human reference genome to adding lots of genomes from different species, adding lots of genomes from different individuals, adding also models specifically for plants, as plant genomics is a bit different. And we also looked at the effect of the model size. So we trained a whole bunch of models from a few million parameters up to 2.5 billion parameters. We had the chance to be able to work with NVIDIA and Google on that. So we got access to lots of compute, to V4 pods at Google, and also to the Cambridge One supercluster in the UK to train the generation of these models. And maybe the first pretty cool insight that you had after training the first generation was to observe that, and would say we have the same kind of observation in protein language models, that despite no supervision, so despite the fact that you just play this game that could be a bit abstract, of you have 6 MERS and you start masking them and reconstructed what's been masked, they start learning about biology. So for instance, in this case, we looked at a model after pre-training. We've never showed this more any annotation. So it's not aware of different genomic segments. It's just been trained through an MLM. And we start looking in the layers, and so we start looking at sequences for which we know annotations and see how they cluster. And so you can see pretty cool effect, which is actually exactly what we expected. To see that in the first layers of the neural net, you start having very simple representations. So basically, it's going to make the difference between the regions of the genomes that are coding for proteins and the ones that are not. And as you start moving down the net and go through the layers, you're going to see like more granular representations, with towards the end of the net, even very granular representation of capturing lots of different genomics elements. If you can look also at the attention maps of the net, you see the same type of behaviors. So here we reused methodology from a protein language models, trying to see if some specific attention, some specific attention head captures some specific elements. We saw that it's the case. Also, with the nice insight that the bigger the net, actually the more focused becomes the attention head. And so you can see, for instance, that in the 2.5 billion parameters, you start having some effects where there is just one specific attention head that specializes completely in detecting one specific type of element, like regulatory elements. And I would say that's kind of the motivation of why you would like to start fine-tuning this network. So they obviously run a representation that at least captures some biology, captures some genomics. And so I would say we create this game that's now been pretty popular in the field and pretty popular in lots of other domains of you pre-train this neural network. And so we can try lots of different data sets for pre-training. In our case, we just reconstruct k-mers, and then you fine-tune. And when you fine-tune, you can do lots of different tasks, classification, regression, with the assumption being that if the representations are good enough, it should be very fast and easy to fine-tune this neural net. In our case, we use the efficient supervised learning, so kind of a LoRa type of technique. And so you move from a self-supervised pre-training that requires hundreds of GPUs for sometimes up to a month or even several months, to a fine-tuning phase, which is like a few minutes on a single GPU, even on downstream tasks where the dataset is pretty large. And probably one of the main contributions of this work has been twofold. So one, we re-explored the effect of different pre-training datasets. So we try to understand what's important. Do you need a genome from different species? Do you rather need diversity from different individuals? And we also worked a lot on building one of the first benchmarks for evaluating these models. So also I mentioned, say that since the different reviews of the paper, this benchmark has evolved quite a lot. Now we have a very stable version with like very recent data. I think the data is at most like 3 years old. So kind of very recent data, high quality data. And the idea was to look at lots of different effects through the genome at lots of different scales, from like understanding epigenetics, which for instance chromatin accessibility, to understanding splicing or gene production in the cell. And we fine-tune our neural nets as well as of other competitors on this test to try to understand different effects. Also mentioned, say that since this work, there's been a lot of other works that have been published. Even at IQR this year, there's been probably 4 or 5 different benchmarks for genomics language model. Pretty creative on that. It's cool to keep seeing the same types of insights observed by people. So it kind of shows that these things are pretty robust. And kind of the key takeaways for us from this work, and kind of nice is that they really echo with what people observe in different fields. So there is, I would say, no surprise. So first, that you observe scaling growth. So increasing the size of the model actually improves performance. Still, I would say a conversation we had this morning is that where you need to go, that's, I would say, still an open question. In the first generation, we went up to 2.5 billion parameters. In the second generation, we showed that we could compress and go up to 500 million parameters and actually increase over the 2.5 billion. We still don't know where to go in terms of size, but I would say at least in the range of few hundreds of million parameters, the bigger the better, usually. In terms of the data, it's true as well. And there, the trend is very clear. The more high quality data you put in the model, the better. But especially the diversity of the data is very important. And so for us, a cool insight, I would say our team is kind of split between 50% of AI scientists and 50% of bioscientists. For the AI folks, it was kind of natural, that the more data you put, the better it's going to work. For part of the biocomp team, I say when we started to explain that we look only at downstream tasks for humans, but that we will start adding genomes from everything you can find, even viruses, they were not really convinced. It was cool to see that it actually helps a lot. And that it's one of the key insights, even if you look at one specific species, in the pre-training set, it helps a lot to add genomes from lots of other species. Still trying to understand why, probably like because of evolution, but yeah, it was an interesting insight. And then we observe like some trends, I would say, in other fields. Architecture is important as well. We started to play, with all this stuff, like enhancement from different literature, like changing the encoding layers, using rope embeddings, changing the activations, removing the biases in the attention, like free connected layer, etc. All these tricks that also kept improving the performance of the model. And finally, we observed that you can use these models for lots of different downstream applications. And I give precise examples in the coming slide. Maybe before going to the next generations of these models, I wanted to spend a few slides on a study that we published kind of end of last year, and I think was kind of one of the requests I got for this talk of saying, can we use these models for proteins? So if you look at proteins, they actually come from RNA, that comes from DNA. So for most of the proteins, at least when they've been synthesized in a cell, we can find the DNA sequence of that protein. And so one could argue, okay, if I have a model that can actually work directly with nucleotide, why would I need a model trained, on amino acid? I could just use that one, compute a representation, and fine-tune that for downstream tasks. So that's kind of the question we try to answer in this study. The paper was called "Are Genomics Language Models All You Need?" There is an interrogation mark. And I would say the main starting point for the paper was to say, okay, we know that for proteins, you have a coding sequence, you have a gene that you can use and feed that to a model. However, if you look at the training data of our genomics language models, we've put full genomes. And for eukaryotic species, especially for humans, we know that the coding regions are only 1.5% of the genome. So these coding regions are only 1.5% of the training data. So it's very small. But not only it's small, but even we're going to have a complete out-of-distribution effect, because in the genome, the exons, which are going to be the coding sequences for proteins, are actually separated by introns. There is alternative spacing. I also say a few words. And so you're never going to see actual full CDS that code for a protein in the genome. And you don't necessarily want to have to find what were the introns, because even because of the isoform spacing, you won't know what they are. So it was an actual question. Is it going to work? And maybe a final variable also is that for proteins, you usually tokenize using amino acids as tokens. So in the case of going to DNA, we know that codons code for amino acids. So we would like to use codons as a tokenization basis. However, these models have been trained with 6 mouse. So you don't have a direct mapping between tokens. So it was, I would say, even a bit pessimistic study, starting saying it's not going to work. We say cool finding was it actually does for some tasks. So we really found when looking at different protein benchmark and fertile, so there are tasks where protein language models are clearly better by a strong margin. There are some tasks where both are going to work, and there are some tasks where actually genomics language models are going to outperform the protein language model. So that was the whole point of the study, trying to understand what's happening. Maybe before going into the details, just an important mention to say the mapping between codons and amino acids is a one-to-one mapping. So you're going to get one amino acid for one codon. It's not true in the other direction. So there are several codons that code for the same amino acid. So if I give you the coding sequence, you can give me the protein sequence. If I give you a protein sequence, you won't be able to give the coding sequence. It's called the codon optimization problem. If you try to take random codons to do that, the performance is going to completely drop. So it's pretty important to have access to the actual coding sequence that was present in the cell. We also looked at other assumptions. Can you permute the order of these different codons? Can you change them but just respect some statistics? We always see a decrease in performance. It's important to use the actual coding sequence that's present in the cell. And so basically, it's a lot of work of doing data curation and being able to have a protein that's been actually expressed in the cell. If it's a purely synthesized protein in the lab, you won't be able to use this type of techniques. Also, another question that you answered in the paper was the importance of tokenization. So we received lots of comments of people saying, oh, you know, your models have been trained with 6 MERS, they're not going to work for proteins, you should at least retrain models on codons directly or on streamers. So we did it. We retrained a full nucleotide transformer using streamers instead of 6 MERS. And it was pretty interesting to see that it actually doesn't really make a difference. So if you look at the performance on the different tasks, for some of them, you gain a bit. For some of them, you lose a bit. In average, actually, you obtain the same performance. So we observe that at least the tokenization is not that important, and that you can use models that have been trained with 6 MERS or even other types of tokenization for this type of task. Maybe before concluding this part, I wanted to deep dive into one specific task. So there is this task of a melting point prediction from proteins that's pretty classic in the field. That's the task where we actually observe that DNA language models outperform protein language models. It's true for both DNA-BERT and NTv2. And so we try to understand why it's the case for this task. And so when we started to chat with our bio colleagues, they said that there are at least two features that they know matter to predict the melting point. So one is the GC content of the coding sequence. And the other one usually is that it's also very related to the species. So I put on this plot like the distribution of a melting point temperature regarding the species. And so you can see that it's pretty easy to separate the different classes. So what we did is that we took both CSM2 and NTv2 and we started to add to the input sequence these different features and see what's the impact on performance. What you observe is that for NTv2, all these features are already contained in the DNA sequence, so adding them explicitly is not going to impact. When you move to ESM, if you start adding the GC content, you start increasing the performance. If you add the species on top, you increase it even more. But you can see that you don't completely close the gap with DNA language models. And so for this task, at this point, we think that these two features are important, and there are probably a few other features that the models are going to be able to capture from the DNA sequence. And to conclude this part, so for now, I've mainly presented an opposition between the two. Should you use one or should you use another? But the answer is why not use both, especially if you have two sequences. So imagine that now for proteins, you have the actual amino acid sequence, you have access to the CDS, and can you maybe put the coding sequence in Nucleotide Transformer, put the amino sequence in ESM or Amplifier or any other protein language model, and combine these informations and fine-tune the two neural networks together. We did that on a lot of tasks. You usually obtain at least the best of both worlds. What was pretty cool was to see that if you were to predict protein stability, not only you get the best performance that you could get with one or the other, but you actually get transfer learning between the two. So if you look at this plot, we did that. We did, in this case, a very simple technique where we just take the embeddings from both models, we concatenate, and we put that in some regression head, and we fine-tune the whole thing end-to-end. And you can see that the joint model has actually a strong improvement over the best of the two, showing that there are probably features that are correlated in both sequences that are analyzed by the model. And so that was kind of the starting point for our second paper that's called the Isoformer that we presented at NeurIPS. That's how we met actually with Quentin in Vancouver, where the idea was to look at this problem in a bit more general way, say, okay, we know that from the central dogma of biology, DNA is going to be transcribed to RNA, that's going to be translated to the proteins. There's been foundation models trained for all of these different modalities. We trained some for DNA, other groups did as well, lots of models for our protein. And recently, there's been also some RNA foundation models. We can also use some nucleotide transformers to do that. And so the question of this study was, say, can we connect these three types of models, and can we fine-tune them together and hopefully get transfer between the different pre-trainings? So to do that, we looked at one specific task that's kind of considered as a very difficult task in the field, which is the prediction of the isoform expression. So there's been several models like the Isoformer or Borzoi that specialize at predicting gene expression. So you're going to give to the model one gene. So it can be hundreds of thousands of nucleotides. And you want the model to predict over different cell types. So for instance, 50 different cell types, how much this gene is going to be expressed. So usually you measure that by, for instance, by the quantity of RNA that you can measure. So how much RNA is going to be actually produced in lots of different cell types by this gene. In this case, we made the task more complex, and so we're not looking at the whole expression of one gene, but we're actually looking at the expression of different isoforms. So we know that one gene is not actually going to yield only to one type of RNA and one type of proteins, but actually the introns between the exons are going to be spaced out in lots of different ways. And so one gene is actually going to produce lots of different proteins and RNA in different quantities. So what we wanted to do here was say, okay, we don't want the total value of the expression for that gene, but we want for this gene the values for all the different isoforms. And so the way we frame the problem is that we build one model where I give as input the DNA sequence, one isoform that could be possible or not, and the corresponding protein of this isoform, and I want to predict across 50 different cell types what are the values that I can expect. And to do that, we connected 3 different encoders and looked at different ways of connecting them. The way that worked best for us was to use this mechanism with cross-attention, where we basically compute cross-attention at almost all the layers of these different models. So you really arrow the information, like the different embeddings to flow everywhere. Now I need to say that we've tried lots of different techniques and we see very small improvement. Actually, at almost the same time. Yeah. Sure. So input is one DNA sequence, an mRNA sequence, and the protein that corresponds to the mRNA. So between these two things, there are lots of repetitions, I would say. And the output is 50 different numbers that are the 50 different expressions for this specific isoform, for this specific gene in 50 different cell types. And so we scraped, so I think the data set comes from encode. And so we did the same that's in Informer. So you remove chromosomes from the training set. And so you train on the training set, and then you look at genes that are on chromosome that you've never seen if you can still predict. Yeah. So actually, it's the Informer paper from DeepMind that did that now a couple of years ago that showed that you can expect that. Here, I would say it's an extension of this paper of not just predicting the, say, total expression of a gene, but going to isoform level. But yeah, I'm happy to share a few resources on that. Sorry. So I was saying that actually at the same time, on most of these studies, there's been this paper by Apple that also looked at what's the best way of combining different encoders. In their case, it was for audio, video, images, and text. And they kind of also show that kind of all the different ways work and lead to the same type of performance. Is also what we observed in this work. Something kind of interesting with this architecture is that you can be very flexible. So as I mentioned, when you look at this definition of foundation models, they can be self-supervised, but they can also be supervised. So the same way that in computer vision, people have built very good encoders by just training them, for instance, to predict classes on ImageNet. People have done that in the field of genomics with Informer or Borzoi. And so you could use these models as encoders. Here, I would say there's no surprise. It's a gene expression task. And Informer and Borzoi have been trained to predict gene expression. So we can use, and I don't have said, but it's the studies in the paper, you could use NTv2 as an encoder for this task, but you get actually even better results if you switch the encoder to Informer or Borzoi, as they've already seen lots of gene expression data. And so probably the nicest result of the paper was to observe that you actually get the transfer learning that you expect between the different pre-trained encoder. So the way we measured that is that we started with Informer for to process the DNA, Nucleotide Transformer to process RNA, and ESM to process the protein. We connected through cross-attention for these models and started to fine-tune to predict this isoform gene expression. We started with all randoms, randomly initialized, and then we started to switch the weights of the different models by actually weights that have been pre-trained with the different techniques. And what we observe is that every time you switch and you replace one of the models from the randomly initialized weights to the pre-trained one, you actually get a boost in performance until the best performance we could get on this benchmark. really showing that strategies where you would pre-train first lots of different foundation models on lots of different modalities, and afterwards start connecting them on data set where the modalities are matched is kind of a viable technique. It's also like the strategy, I think, of lots of startups. There's been a GenBio recently that's, I think, going in that direction. There is Bioptimus and Okin and other like companies doing these kind of things. So it was cool to see that it's actually probably a good way to address this problem. I would like now to show something a bit different. So when we started, and I would say like lots of other groups, to look at this problem, we made a strong parallel between genomics and language, saying actually DNA can be seen as the language of life. You could see chimeras or motifs in DNA as words, genomes or genes as sentences. You could use of these techniques like an next token prediction or mask language modeling, and they would make sense. But then could we maybe, instead of looking at this problem from an NLP perspective, look at this problem from a computer vision perspective? And in computer vision, there's been recently very cool foundation models, such as SAM, SegmentNT from Facebook, that have been developed, where they show that by grounding the model into the world through segmentation, so in this case, you take an image and you try to recognize of the different elements of an image, you can build very strong foundation models. However, at least I would say one of the accepted understanding of genomics right now is that genomics is DNA is maybe also very structured, like an image with different portions that have different rows. There are now lots of very curated annotations. You know that some portions are genes, some are not. You know that there are regulatory elements such as enhancers, promoters, silencers. You know that in the gene structures is going to be exon, introns, splice sites, etc. And so a question we asked was, could we use these techniques to train foundation models, but instead of using techniques from NLP, actually to use techniques of computer vision by learning to segment the genome using these annotations. And so to do that, we scraped a lot of data. So we started by scraping 2.5 million annotations. So these are annotations that have been curated by lots of different labs. Usually they use lots of different assays that they merge together to annotate the genome. And so we looked at 14 different elements, genic elements, regulatory elements, and we started from the nucleotide transformer that's been pre-trained. And can we now keep training this model, but through doing segmentation? And so we put on top of this nucleotide transformer a 1D unit, which is kind of the state of the art for segmentation in images, and started to fine-tune the model on all of these annotations. The first, I would say, cool result was to observe that you already get transfer between the first pre-training phase with mask language modeling and the second training phase where you start learning to detect these annotations. If you just take the unit without the big transformer before, you get it's hard to get some decent performance. You can scale this unit up, start increasing. You put a big transformer in addition of the units, it helps a bit, and then you pre-train the transformer with mask language modeling, and there you see a huge boost. And actually, for us, it was pretty cool because on many downstream tasks, you usually ask for ASM. I would say you see some boost between, when you pre-train or not and you fine-tune, but it's rare to see a boost where you multiply the performance by almost 3, which was the case for this task. Another cool insight is that in the community, people have been very interested in segmenting the genome for a while. For instance, regulatory elements. The way it's been done so far, for instance, in the case of an answer, is that an answer is a sequence of 200, 300 base pairs. People create data sets where they put lots of these sequences that they know are answers. Then they create decoys or sequences that they expect are not, train a binary classifier, and then use that to detect in the genome elements. When you look at these papers, and actually we also play this game in AT, so we are also guilty, everybody report accuracies of almost 99%. So it looks like they are extremely performant. When you actually slide this over the genome, the performance drops completely. Whereas if you actually treat the problem as a segmentation and you directly run to segment from the genome, you get a boost in performance. You get actually even a larger boost from on other elements. Some other interesting insights that we know that for DNA, lots of effects can be what's called distal. So they can come from regions that are far away in the genome. That's why now I would say there is a race of making this this model is like taking larger and larger context. In this case, we took the nucleotide transformer that could go up to 12,000 base pairs and use different interpolation techniques to kind of increase the size of the input. We managed to train it up to 30 kb, and we kind of kept seeing an improvement in performance when we increased the sequence length. But then we even like performed zero shot and went up to 50 kb and kind of kept seeing an improvement. To give you an example of what it looks like, so on that slide, the input is a sequence of 50,000 nucleotides. So I couldn't represent the letters on the slide because it would be too small. So you have 50,000 letters that enter in the neural net, and you're going to make 14 predictions per nucleotide. So the way we frame the model is through instant segmentation. So we make the assumption that one nucleotide can belong to several elements. And so for each element, you're going to return a probability between 0 and 1 of belonging or not to this element. On that example, I took a sequence from the test set where there are 3 different genes in the sequence. And so we splitted like all the different plots just for the sake of visibility, but you get all of them in one go. So you get, in this case, you get 700,000 numbers in one pass of the neural net. So that's a lot of information to process. And you can see, for instance, that the model can capture the different genes. It can capture also the genic structure, like with the exon introns structures. It can capture the splice sites, regulatory elements, and so forth. And so now that's typically a model we use a lot internally to screen mutations of patients and try to understand, for instance, the case of cancer, splicing is very important. You want to make sure that you're not going to have a change splicing in a given cell. Kind of to conclude this one, we also run cool studies. So one was to take this model that had been trained only on the human genome, so only on annotations from this human genome, and see if we can in zero shot annotate the genome from other species. So actually, we scraped 16 different species for which we had high quality annotations. And so we just trained on human and evaluated. I would say no surprise, even if it's a bit bad for the ego, but it works very well on gorilla and macaque. Maybe another one that was less expected, but pretty fun. The model is extremely good with beavers. Actually, the performance is better on beaver than on humans. So if there is any beaver geneticist in the room, you can use that for sure. And also, as you can expect, if you go to organisms that are very far from human, you start seeing a decrease in performance. Then you can keep training the model on more and more species. So the way we evaluate that is that we're very careful. So for each species, we separate like training and testing chromosomes. We also make sure we know that some genes are conserved between the different species. So we remove these guys just to make sure that we're not learning by heart the genes that are present everywhere. And then for species, we also have training and testing species. So then you can ask questions like, if I start adding species to my training set, am I going to become better on their testing chromosomes? But am I also going to get better at other species? And kind of the answer is yes in both cases. So by adding species, you become better for this specific species. But also the more species you add in the mix, the better you get at everything. So it's still the same kind of effect that you observe in NT, that diversity is very important, and that there is lots of transfer learning between the different species. So now kind of for the final part of this talk, I wanted to show something kind of different, where we asked this question of, can we give a voice to the nucleotide transformer in some sort? And kind of the idea behind this work was say, okay, so far we had this model and we've been fine-tuning it for lots of different tasks. So we had 18 different tasks in the first paper. People have been fine-tuned this model for lots of other tasks. And so now there are fine-tuned versions for lots of different data sets. However, we know that lots of these data sets are related, right? For instance, there have been lots of these versions of the model that have been trying to detect lots of different types of histone marks. They're all related. Now we have one model per type of histone. Question was, can we build one model that can link all these data sets? So can we get transfer learning at downstream task time? And in this work, the assumption we made was, can we achieve that through English? Can we make the model conversational? And in this case, start getting transfer learning between the genomic concepts in the sequence and the word that's related. So can the model link the concept of splice site to a splice site? can the model link the concept of promoter to a promoter, and so forth. There are different motivations, maybe two main motivations for this work. The first, that we wanted to get an agent because we also want this model to be used by our BioNTech colleagues. They don't all have a computer science background. They don't know how to code. Some of them are wet lab scientists, so they are not going to use a notebook and to fine-tune the model or to compute probabilities. So if you can make this model an agent and actually, just give them sort of a ChatGPT where they can ask questions, put their RNA-seq data, put their FASTA files, and start asking questions about the data is going to be much easier for them and also for the people in the community. And the second, also, I would say, more scientific interest was to see, can we get this transfer learning? Can we use English as one possible medium to connect concepts in biology? To do this, we use techniques from computer vision. So the same way there's been now lots of multimodal image language mode that have been developed, where usually you put an image, you use some encoder like CRIP, you get embeddings of this image, you project that some way in the latent space of a big language model, and you decode, and so you can ask a question about an image. That's in the case of DNA. So we have a DNA sequence that flows in the nucleotide transformer. We get embeddings. We use some neural network, in this case a perceiver resampler, to project these embeddings into the latent space of some language model. In the paper, I think right now it's Llama 1. Recently we updated to Llama 3, 400 billion parameters. And then you can ask some question and train the network to answer the question. The way we did it is that we didn't want to touch the large language model, mainly for compute reasons, because it's not fun to train a model with 400 billion parameters. So we restricted to train the projection layer and the nucleotide transformer. So if you want to run some kind of soft prompting of the large language model, and so you expect that it's going to run to answer the different questions. We created a data set, I would say manually, or almost manually. So we scraped the task from lots of different papers, including ours and other groups. We ended up with 30 different tasks from the researcher. And for each of these tasks, we worked with our colleagues to write examples of questions and answers. So it can be questions like, is that sequence going to be methylated in this cell type? An answer saying yes, no, with details, etc. We did that for us of this effect. We also reuse the task from the paper that I showed on proteins. So in this case, we give the CDS, and you can ask question, how can you compute the melting point of this protein? How can you tell me what the fitness of this protein is function, and so forth. We kind of try to create a combinatorial. So for each task, we created a bunch of questions, maybe 20 questions, 20 answers. So you can already have 400 possible pairs for one sequence, and then you also have the combinator of the sequence. So in the end, you have a data set that's pretty diverse to train the model. The first pretty cool result was to observe that by doing this, you indeed get the transfer that we were looking for. So here I'm looking at the 18 tasks from the Nucleotide Transformer paper. Each of these points is one fine-tuned model of NT. So you take NT and you fine-tune for each of those tasks, whereas these purple points are always the same ChatNT model. So here to move from one point to another, instead of fine-tuning a new model, a new data set, I just change the question. I ask, can you find any presence of histone modification H4? Or I ask, can you find any data promoter in this sequence? And then I get the output of my neural net. Something interesting that we were a bit scared in the beginning that the neural net, could answer some things that are completely irrelevant. It could give you the recipe of a cake, whereas you ask for a presence of an element. After a few gradients, it's not the case. So it's very quick. You train for maybe 2 minutes and you start producing answers that look good. They're wrong, but they all look good. And then you train for hours or days, and they still look good, but they become accurate. And at some point, in terms of pure accuracy, so you just extract from the answer the actual binary answer, for instance, yes or no, or the value in case of regression, and you compare. You already see that you start increasing the state of the art. And notably, we saw an increase in all these histone mark tasks. And the assumption behind what is started to actually connect the concept of histone and start reusing motifs and different features. I don't have a slide on that, but we're going to release very soon. We have a new version of the paper where we use that gradient attribution techniques to see like what's doing between what's being predicted in English and in the DNA sequence. And you observe like the types of things that you're looking for. So in the case of the data promoter, you see, for instance, that if you backpropagate the gradient from the English, you're going to find the data box. Same for specialized junctions and others. So we really see a connection between the English concept and the genomics features. We did that for lots of different tasks. You can have a lot of fun, can do that for classification, regression. In the case of regression, we asked the neural net to write in English the numbers. It sounds like it could be not the best idea. It works. It was pretty interesting. For instance, I talked before about like melting point prediction for proteins. In this case, you can ask a ChatNT to give you the melting point of a protein given a CDS, and you can see that if you extract the number from the predictions, you get a very strong correlation between the prediction and observed value on the test set. We did that across 30 different tasks for a paper, and it's always the same model for all of these different tasks. And maybe to finish this presentation, one last thing that we wanted to look at was to try to understand what's the confidence of this model. So the first time we presented that to our BioNTech colleagues, they were all very scared, saying we've all played with ChatGPT, we all know it hallucinates a lot. Oh, you know, are we going to make sure that your model is not going to hallucinate? So our first answer was, all deep learning model hallucinate, you know, even if it's not a language, it can give you wrong predictions. So guys say, yeah, that's true, but if I have a classifier, I can, you know, at least look at the probabilities. I can maybe calibrate my classifier, and at least I have some confidence, you know. And if I see that the confidence is too low, I'm not going to give it a try. They're like, whereas your model, if you just ask a question, it returns something in English, I have no idea of the confidence. You know, maybe it could be completely wrong. Maybe the model could be very confident. So that's something we wanted to look at also in the paper, was can we estimate the confidence of the model? And so to do that, we're going to select one question. Can you find a promoter in this sequence? We're going to generate a few examples for both the no answer and the yes answer. For instance, five of each. And then we're going to compute the perplexity. So perplexity conditioned on the DNA sequence. So we compute the perplexity of the large language models for all of the negative answers and for all of the positive answers. We average that. And so we assume that the average of the perplexity can be interpreted as a logit. So now I have a positive logit and a negative one. I can feed that through a softmax layer. And so now I have two probabilities. And I can actually look at the calibration of the model and at the confidence. What we observe is that the model, like by default, is not so badly calibrated. Actually, you can interpret some of the confidence, but then you can use other techniques, for instance, from scikit-learn, like you can feed a Plat model or other techniques, and you can calibrate this more. And so we show that if you have very case-sensitive applications, you can afterward fine-tune the model and calibrate it for that application. And to conclude this presentation, just a small idea of what it can look like. So I cannot demo it, but internally we actually have a kind of a ChatGPT product where we have this model, so scientists can connect and then start adding FASTA files, RNA-seq, ask their questions. And so you have something very interactive like that, where you can ask different questions, and then we show like you can put also different sequences, move from proteins to DNA, etc. And to conclude this talk, mention that most of these works are open source. We didn't read the training code, but we read all the checkpoints. Most of the data was pretty cool for us to see that this model got pretty popular. I think now we are at a bit more than 1 million downloads actually this month over the different nucleotide transformer models. So they've been used a lot. We also read SegmentNT, the versions of the model functions for proteins and so forth. And I would like also to thank the different colleagues. I think it's just a subset of the people. Since then, we had the chance to work with many other colleagues across BioNTech, Cornetech, paper on proteins was done with MIT, also NVIDIA and Google partner. Right? Thank you so much. Happy to take any questions.
Great. So I think we have time for a few questions from the audience. Anybody here? All right.
Who's first?
Thank you, Thomas, for the great presentation. I have two questions related to the same topic. What is the adoption of this chatbot at BioNTech? And the second one is, it seems to me that the question must be specific. Like, is there an answer with this within this sequence? Do you expect in the future to have an agent who is capable of finding what is wrong in some specific sequence?
That's a very good question. So I would say what I showed this ChatNT is, I would say, only a subpart of the agent that we actually released. So we're looking at lots of different applications. So actually, I would say like other companies, the same agent can also, for instance, go scrape the data internally, go perform data analysis, scrape research papers, give you summaries, things like that. I would say these different capabilities are much more used right now. There is also a Turing function in the bot, so you can try to use as ChatNT, put the sequence and directly ask questions. You can also ask the agent to use SegmentNT, for instance, to run it over a sequence, analyze the results, plot them, and start reasoning. I would say, and we call this agent a Lyra internally, in a reference to Star Wars. We are more using, I would say, this one. The ChatNT is more, I would say, research at this point. To answer a second question, I would say that's what we expect one day. It's not the case right now. Right now, it's going to work in the realm of the different tasks that it's been trained on. What you observe is that it's robust to the way you ask the question. So you don't need to ask the question in a predefined way. It does not need to be one of the questions of the train set. So you can, and actually in the test set, we completely change the questions. And the way it answers also changes, even if the accuracy does not move. So that was, I would say, the first evidence we saw. If you get a completely new task, it's not going to work for sure. What we hope is that one day, if you scale it enough, you start seeing like a generalization to new task.
Hi, thanks for the talks. For the talk, can you go back to the slide with the baboon and the humans, where you show the different metrics across animals? I was just wondering if you played around with these results, trying to show some kind of like a correlation with our current theory of evolution, showing that there might be some flaws in our actual theory of evolution, by just saying that there is actually a much closer relation between like human and cats, for example, that we think there is according to the theory of evolution.
Yeah, no, it's a good question. I would say we haven't explored that too much. For us, we are mainly interested in human. I would say we did this experiment as a sanity check for a paper. Also because part of the people in the team have a PhD in evolutionary genomics, so they wanted to know. We did not have the luxury to spend too much time studying that in depth, but it could be a very cool follow-up. Yeah.
Thanks. And also, second question I had, it's much more technical, but the first transformer you introduced, you decided to consider tokens of 6 nucleotides.
Yeah.
And I was wondering why such a design choice? Why not 3? Because as you said, 3 is like how we encode.
So basically, the design choice came from 2 constraints. So in DNA, as mentioned, you want to take as long sequences as possible. So in the first versions, we managed to go up to 12 kbp. It's actually too small. So now people are working more to go to 1 million. If you want to be able to analyze all the human genes in their, like the full genes, if you take like the introns can be very long, what I showed. There can be regulatory elements like enhancers that can be very far. So usually people agree that with 1 million you can see everything. In this first architecture, the idea was just to say, oh, let's try to go as large as we can. So we wanted to put as many nucleotides as we can per token, right? Because as we use a transformer, it's quadratic scaling. So you cannot increase indefinitely the number of tokens. So you need tokens that are as expressive as you can. We tried to train tokenizers using BPE, these kind of things. We didn't manage to get any good compression. And also afterwards, if you have tasks where you want nucleotide resolution, like a SegmentNT, if you use the BPE, it's really not fun. Because then you need to know afterwards how many nucleotides were in each tokens and so forth. So it was easier to use a constant number. Then when you're going to start increasing the number of nucleotides you put in each token, you're going to increase the vocabulary size of the neural net. And so you're going to increase the number of neurons on the final layer. At some point it grows. And so 6 was kind of a trade-off between going like bigger in terms of sequence without completely blowing the final layer.
Okay, thanks.
Hi, thank you for the talk. On the same line, when you have these 6 MER, so you go 6 times over each sequence to have it slightly shifted or not?
No, it's a non-overlapping.
Yeah, okay, but so since what makes sense is
when you train, you mean? Oh,
yes, when you train, let's say you have your sequence, what makes sense is the 3 MERs, if you consider them and you start at the right point and you end at the right point, right? So if you shift it slightly, your 6 MER, you're not gonna get the information from the actual units that make biological sense.
So you mean in the specific case of the protein task, or in general?
In general, but I see like on your scheme, on the top
in
right.
In general, the notion of codons, like the streamers, makes only sense in the coding regions. Okay. Outside of that, there is no notion of codons. So that's why we also investigated that. So encoding regions, and we know in coding regions where the codons start. So when we look at these models trained for proteins on coding regions, we make start the 6 MER where the codons start, and we have 2 codons per 6 MER. When you train on full genomes, you have no idea of what is what. You have no referential. So you just take a random portion somewhere and you tokenize. What we show is that, so what you write is that there are 6 different possibilities, right, for each sequence because you can shift. We train for a long time, so it's going to see lots of different possibilities because we just sample randomly and tokenize. What we show at testing time is that it does not depend on the referential. You can like shift everything by a few nucleotides and shift all the tokens. We get the same performance. Yeah, definitely. Yeah.
I do have a few questions. Oh, go for it. Go for it. Oh, yeah. More. Thanks. That was super interesting. Also, the whole talk. I had a question about your the training set you used for the first LLM. I saw that you used the 1000 genome project data set, and I was wondering if you use the full sequences of this, because also like I think the power of this data set is in the few positions that can make differences across ethnicities, and if you put the whole sequence, maybe you sort of there's a lot of redundancy in these sequences. And so yeah.
Yeah, no, exactly. Thanks for the question. Makes a lot of sense. So yeah, in this, I would say in this model, we had a bit this naive view where we just wanted to look at genomic sequences, so just cut genomes. The way we trained on the 1000 genomes was we took the human reference genome, we took the VCF files of the 1000 patients, we would sample randomly a region in the human ref, randomly sample one patient, look at the mutations, and force them in the sequence and do MLM on that. So indeed, exactly what you mentioned, what you observe is that it's very tiny changes, right? Usually it's 1 letter if you're at 1000 tokens. So we think that's why actually when we trained on that, we didn't see an increase in performance by adding the genomes of patients. I would say there's hasn't been yet a work that we managed to exploit these mutations. It's one of the things that we're working in right now. 1000 genomes is one way of doing that. There is also NTEX and TCGA, where you don't only have the genomes, but you actually have also the results of lots of assays. So you really try to understand how these mutations are going to impact RNA production in different cell types. It's, I think, where there is the biggest avenue for improvement, and so we're looking at that right now. Mm-hmm. But yeah, this more is not very. It can detect some types of mutations that are actually studied in the paper. It's not super good at that. There are lots of mutations that are going to be smoothed out by the model.
It's a really interesting concept though to try to include that, and yeah, for future work, I think there's a lot of interesting things that can be done with that. And also, is there a reason why edible plants
Yeah.
credit?
There is a reason, because we have customers in the agriculture space that are interested into modifying maize or soy or other plants. It's also a cool problem in itself. I would say there is food security things. So we actually worked with NVIDIA on the human genomics and with Google on the plant. So there are 2 papers. Many talked about the Nucleotide Transformer. There is a paper called AgroNT, where we train on these edible plants. And we show that the genomics is pretty different. So the types of tasks that I look at are pretty different. We use the same techniques though, and we show it still work. But so yeah, for us, even in terms of team, it's a bit 2 different teams. I mainly work on the human part, but the plant part is fun and it's something where we're doing business also.
À ta droite, puisque tu as mentionné apprécier parler en français, je voulais juste te remercier de ta présentation. I'm going to switch in English for the question. You mentioned, I think, here that you used 800 genomes for the training. I was just wondering, have you made any choice about which genome do you pick, or it was just data availability that was the decision on?
So yeah, the decision in this case was called Mary, who is my colleague, who's geneticist. She looked at all the genomes. We had the maximum data base size regarding the compute we had. She basically tried to balance a bit the tree of life. And so she handpicked. To be fair, we didn't perform an actual study. She handpicked herself genomes to try to balance. So she wanted to have mammals, she wanted to have plants, viruses, bacteria, and so she kind of tried to balance all of them. Also knowing that they have very different lengths. So there can be genomes that are very long, some are very short. So she tried to balance the diversity.
And following the lunch talk that we had today, do you think there was there is a probable good choice to do to filter out some genomes that are not useful?
It's a good question. It's something that we still want to investigate. I would say the problem is that the compute cost of these things is pretty high. So it's a hard question to investigate at the pre-training choice. We also discussed with several groups. So there are lots of different things you can look at. You can choose what genomes you show, but these genomes, you can also process them and clean them. We know that there are regions, that appear a lot between different genomes. We know there are regions that are repeats, that are maybe less interesting. So you can also perform lots of data cleaning on all of these things. There are some groups that show that on smaller models, it has a very important impact. In our case, it's not something we have explored too much for now. We are a bit more in this mindset of saying, okay, we've pre-trained on this multi-species. We know it works to some extent. And now we are more working on adding other modalities to the mix. Though I wouldn't be surprised that some group work on that and show that you can increase further the performance by doing this. Yeah.
And very last quick naive question on your Chisney first slide. So I'm not going to ask you to go to the first slide, but it's here. We see for the antron and intergenic region that there is probably a small cluster between the two. Have you investigated what is it, these regions?
I can ask.
Okay.
I couldn't tell you. Yeah.
I was just curious to see this in the middle, but never mind.
Moi, j'ai pas de contraintes.
Oui, ça va, okay. Okay, but parfait. Well, thank you very much. First of all, good audience to reveal that the beaver DNA genome is similar to humans, so you're in the right country for that, by the way. My question, the core question I had was about all the interpretability that actually you're exposing with this slide here. Actually, that's then you could investigate the attention layers to figure out if there's any specificity to specific interactions between regulatory elements, for example. Have you tried exploring that kind of direction for the let's say Isoformer work, where the cross attention mediated between modalities could reveal how, let's say splicing variants give rise specifically to protein sequences, even if we know that a priori, do you think it's worthwhile to investigate that within the model's cross attention?
Yeah, definitely. So yeah, I think it was right. We've tried. We didn't manage to get any interesting insight, I would say, so far. I think in the paper we might have a few attention maps where we show some effect. So far, we were not able to capture really interesting effects in the Isoformer. I would say also because of the combinatorial of all the different cross attentions. is still something that we would like to explore for sure.
And for the Isoformer work, do you think something like contrastive learning would be helpful to kind of bind the representations
to
together?
align the different representations before. Yeah, I think it could be interesting. Yeah.
Okay, that's all I have. Well, I have more, but I think that's fine.
Yeah, well, in the interest of time, thank you everybody for your questions. Let's take another moment to thank our speaker. And that finalizes the session for today. Thank you, everybody.